Potential of high-throughput confocal microscopy, multidimensional and multimodal analysis.
Okay, so thanks for the introduction and for the invitation to speak today. So I'll be talking about some of the image and data analysis challenges that we face at AstraZeneca. I'll be framing this around the high-throughput confocal imaging platform that we've recently started using. So the work at AstraZeneca spans the whole range of medicine development, the whole timeline. And I'm in a department called quantitative biology, and we tend to focus at the very early stages of drug discovery and identifying potential drugs. So we aim to use quantitative and statistical techniques to improve the prediction for what will be a successful drug during the clinical trial phases. And as we've already heard today, the drug attrition at these stages is a major challenge, and so we can have a big impact on the development of new medicines by making better predictions for this stage. So the quantitative biology department is made up of a range of disciplines, from bioinformaticians to mathematical modelers, image analysts such as myself, and as well as statisticians. And we have a wide and diverse range of backgrounds as well. So my own background is an academic one, and so in my academic background I've been focusing on the extraction of information from time-lapse movies. I won't go into any details here. This is ranged from looking at cell division, gene expression, and cell fate choices in stem cells. But today I'm going to be talking about our efforts to make the most of this high-throughput confocal platform. And so this plate-based assays are kind of the main workhorse of high-content biology, and this allows us to, in a single dish, to be able to test hundreds of potential drugs, hundreds of potential combinations of drugs, or different cell types and different cell treatments. And adding to this a spinning disk confocal unit improves the resolution of our imaging, particularly in the Z direction, so in three-dimensional imaging, and allows us the potential for single molecule detection. And also the spinning disk confocal is also much more gentle on the cells, so less damaging during the imaging, and so this opens up the possibility of imaging the cells live and getting kinetic and dynamic information from this as well. So I'll just go through some of the capabilities of this machine to illustrate the challenges that we face. So from the motorized stage, you can capture multiple fields of view within a single one of these wells, and so potentially imaging every cell within one of these in a well, so potentially building up the whole population of cell information. For a single well, we also can look at multiple fluorescence modalities, so labeling different proteins or cellular compartments with different wavelengths, and then combining this to form sort of multi-channel images. And then there's also the depth information, so we can interrogate multiple slices and looking at three-dimensional cell culture systems. Added to this, we can also then look at the kinetic and dynamic information, and there's two ways that we can go about this. So we can focus on a particular well and capture a burst of images and capture very fast dynamics, so receptor internalization, vesicle dynamics. Or we can capture the whole plate at predetermined intervals to follow cell processes evolving over time. And there's also the possibility of combining these two and imaging at several different imaging rates. So for instance, if we're interested in cell division events, we might want to image at faster intervals, but we also simultaneously want to look at cell fate choices which occur over longer periods of time. And so we can also then combine these two different dynamics. So this gives us a huge amount of complexity in the experimental acquisition, which is reflected in the complexity of the images which come out. And so as well as the size of the data, there's also the organization of the data. And so now I'll just talk briefly about some of our recent attempts to actually parse and to handle this data. And so what we've been building is a software platform which will allow us to handle all these different possibilities of the imaging, and to be able to interactively browse the huge imaging data sets that we're building. And combined with this, we can then visualize this in 2 dimensions, 3 dimensions, or 4 or greater dimensions, to be able to interactively look at the data that we're collecting. And the idea behind this is to build a platform which will provide a consistent interface for manual interaction, but will still be amenable to high throughput analysis. And so to do this, we're using an object-oriented programming approach, whereby the idea is this modular approach where we can plug in different experimental setups and plug in different visualization tools into this consistent framework, which will allow us to quickly get on top of different experimental acquisition setups. And we're further developing this to be able to look at to plug in the downstream data analysis into this platform as well, and to be able to extract high throughput information, high throughput data using the consistent interface. And this will allow us to then have easy quality control for manual checking at the end to ensure that the data that we're getting out is reasonable. And so we're basically building this kind of high throughput workflow. So I've shown you some of the image visualization tools that we've got, and the idea is that this will plug into a workflow including the segmentation of images, the subsequent measurement of images, and followed by the export into multi-dimensional data sets that will then be amenable to further downstream analysis, but also quality control images, incorporating the segmentation and measurements for fast user verification as well. And the hope here is that by building this in this modular way, that we can interrogate 3-dimensional, 4- or 5-dimensional data sets in the same way that already exists for the 2-dimensional high throughput imaging approaches. And so one example of what we've already been doing with this, so previously we've been looking at 3D spheroids. So this is a 3D culture whereby cells cluster and organize into a 3D structure which bears more characteristics of the real in vivo system than does a simple 2-dimensional culture of single cells. So previously we were looking at a simple 2-dimensional analysis of this, and through this new framework we're now looking at interrogating the full 3-dimensional structure and looking at the cellular organization within these spheroid structures. So I'll go on now to talk about some of the challenges that we face in doing this. So some of these are in terms of the new modalities that this platform presents, and one of these is called digital phase contrast. And so this is an interesting approach which essentially calculates the optical thickness of cells, and so allows you to visualize cells in a label-free manner and to potentially segment these. And so this, because it's label-free and non-damaging to the cells, it presents possible opportunities for looking at cell shape, cell texture, and so there's the potential here to replace multiple fluorescent stains and multiple assays for cell health, cell proliferation in this label-free and manner, which we can use in a time-lapse approach. So there's the potential here to gain more understanding of this approach and potentially simplify and gain simplify the approach and gain a lot more information from these sorts of images. And so the other kinds of challenges we face relate to scaling up traditional assays into a more high throughput manner. So if we think about time-lapse challenges, which is my background, we need to link cells to be able to track them over time. But we also need to think about what happens when you have errors in the tracking, and can we come up with an analysis which is tolerant to these errors of if there's a tracking problem. How can we develop an analysis which is robust to these kinds of these errors, and be sure that a change in the appearance of the cells doesn't induce a change in the tracking accuracy, which then causes a change in our interpretation of the data. So we need new methods to be able to scale this up to a high throughput approach. And with time-lapse imaging, we also get more information. So we also, if you track the dynamics of this the cell intensity over time, rather than just a single readout, you now get a whole vector of values. And so we need to think about what's the best way of interpreting this additional information that comes out of this. And we also face, I think, like many people, this explosion of data that we're now able to acquire. So our ability to acquire the data has vastly outstripped our ability to interpret it. So now when we image a well, we can get many measurements per image, or even per cell if we're making single cell multiparametric measurements. And so what's the correct way of actually interpreting all of this information that comes out, so cell shapes, cell textures? And so often this data is very highly redundant, so measurements will be highly correlated. So we need to think about ways that we can correctly interpret this vast amount of data, and think about how errors, for instance in tracking, or for instance in cell segmentation, how do these errors propagate through to the data, and how does that influence the conclusions that we make from it? And also, what effects do the choice of analysis that we take, the particular algorithm that we choose, for instance in dimensionality reduction or multivariate analysis, what effect does this have on how we interpret the experiment? So this is something that I think at the moment is unexplored. So I think there's also great opportunities for machine learning here. So there's a couple of ways that we can think about this in terms of classifying images as a whole, or in terms of helping us to segment the object within an image. And I think we've seen a couple of examples of each of these today as well. So I think there's an awful lot of opportunities for incorporating these machine approaches to help us interpret this data that we're generating now. because this amount of data is only going to increase further and further. So we're looking at now confocal imaging, but already there's the possibility of using, for instance, a lattice light sheet microscope, which will generate even more data at faster temporal resolution, and other light sheet methods which will allow us to look at whole organisms or whole organs. And related to this, we're still using these kind of 2D cell culture paradigm, and there's the potential to transition to looking at three-dimensional cell culture systems and which contain more relevant information. And already we're starting to think about how to build kind of tissues on chips. And I think this presents a lot of opportunities for the kind of multimodal imaging that we've heard about today. For instance, combining fluorescence imaging with mass spectrometry imaging and other metabolic approaches. And so I think there's a lot of challenges in combining all this data, but I think there's a lot of benefits to be gained from doing that. So I think I'll stop there. And yes, I'd like to acknowledge especially Yin-Hai, who's in the audience today as well. Yeah, and I'd like to thank you for your attention as well. Thank you.
Thank you very much. Questions?
When you described your software, I saw a big GitHub on your slide. Is this something you're open sourcing, or?
It's in our plans to do that, because I think it will help in terms of the development and making a more universal approach to kind of handling this. We're not doing that yet, but I think once the platform is a bit more mature, that's something that we would definitely want to be doing.
Your cells, when you track them, what do they do? Do they undergo topological changes?
Yeah.
And stuff like that. So okay, yeah.
Yeah. So I think the challenges there really depend on the frequency of imaging. And so if we're wanting to be able to scan an entire 384 different wells simultaneously, then necessarily we can only image each well every 4 or 5 minutes. And so there's a potential there for cells to move and change shape. So I think that the approach that we'll have to take there won't be sort of computer vision approaches of object tracking, but we may have to link objects from frame to frame. So I think there may be some challenges there.
With your folder with images set overnight, you had 110 gigs. How long does the measurement actually take, and what is the major cost of taking so long?
The measurement of
the imaging scanning.
So I guess it depends on the type of imaging that you're doing. I mean, in this case, I think this wasn't even as much as could be captured. Essentially, if you're trying to get single molecule precision, then you'll need to image at fairly long exposures, and so this is a limitation in how long you can capture. If you're wanting to, it depends on the process that you're wanting to image as well, because if you're looking at cell differentiation and cell fate choices, you may then need to image for a lot longer, so 48 hours, 72 hours, in order to get an idea of what the end point is. So I think there's lots of ways where the amount of data generated can really snowball. And so I think specifically the temporal resolution is something where faster temporal imaging is always better, but that comes at the cost of vastly increased data generation.
With the cell fate studies, are you tracking single cells undergoing a differentiation, or are you tracking on a sort of more population level? And then if it's the latter, how are you dealing with the heterogeneity in the populations?
So, I mean, previously we have done it in a population level manner. But what we want to be able to do is to look at the final differentiation status of cells and then track that back to see what has happened in the history of that cell, and to be able to identify differences in cell behaviour at the earlier points. So I think that's something which is still ongoing, but I think our aim is to be able to track cells through the duration of that.
Any more questions? If not, let's thank Adam again. Thank you.